We'll again thank you for everyone for online, offline crowds for all 12 countries. We're excited to have you, and we'll begin our lesson today. So before we delve into the full lesson, the quick outline is that we'll start off with a case study, and it's a new format, but we'll start looking at like a medical case study. Then we'll look into how this leads into the field of organ engineering, and then we'll look at bioprinting as a subset of organ engineering. And that's important to remember conceptually, that we have organ engineering as the overall field here, and a subset of that, a tool we can use within organ engineering is bioprinting. A tool under that, which we can use to help bioprinting, is BioInk. So this hierarchy of what's biggest to what's smallest is important, and we'll kind of get into this a little bit later. But without further ado, let's get into our lesson. So before we discuss the specifics of organ engineering, let's walk through it. Let's walk through the case of Mia. So Mia is, let me get the pointer over here. So Mia is a young pediatric patient who has polycystic kidney disease. Now, many of you may not be familiar with this kind of disease, but it's genetically inherited. And essentially what happens is that you have cysts that grow all over your kidney. You have these fluid-filled sacs that grow all over your kidney. And this disease is progressively worse over time. So what happens is that these cysts essentially occupy space in the kidney, stop the kidney from being able to filter blood properly, and that leads to blood being left in your urine. So one day, Mia was just playing. She was 16, 17 years old, just a normal high school student, and she noticed abdominal pain. She had some blood in her urine, got really concerned, and had to go to the hospital. And as a result of this trip, what the hospital told her was that, yeah, you're going to need a new kidney. And many of you may not know this, but the process of getting an organ transplant, specifically in the U.S., is extremely, extremely difficult. It relies almost entirely on people that are that donate or are benevolent and can donate their organs to you, and it relies almost entirely on fate and what can happen. So it's a very, very difficult kind of process. So obviously, I mean, I'm sure many of you are 16, 17 years old in that age range. Can you imagine going every single day, multiple times a day, and best case maybe a few times a week to the hospital to get dialysis? It's just an excruciatingly painful process. It's bad for quality of life, and it's just difficult overall. And one thing to note is that this condition does get worse over time. So as she ages, her kidney function just gets worse and worse, and she just progressively gets in more pain. So then we have to ask ourselves as scientists, is there a better way to go and deal with this issue? And that's where the field of organ engineering comes in. So in the status quo, we have to wait. Patients like Mia have to wait for someone else to donate a kidney. Patients like Mia are reliant on fate and all the bureaucracy that goes on in the legal system to be matched to be matched with a new kidney. And organ engineering is kind of there to alleviate that process. So before we kind of go into the specifics of how organ engineering works, essentially this is just a scope in the U.S. alone of how many people need a kidney transplant versus other kinds of transplants. So surprisingly, it's one of the most needed organs, which is why I chose to use it as one of our initial case studies. So at the higher level, and I'm sure you all are familiar with the way I like to teach, I'm going to give you the overall process, a very zoomed out higher level picture, and then we'll go and start picking it apart. So at the higher level, how does organ engineering work? What we're doing is we're taking a defunct organ. So this is a kidney. This is a diagram representation of a kidney. This is a kidney that doesn't work, right? The cells in the kidney have ceased to work. Then we're going to decellularize it. What does this mean? At the higher level, what decellularizing it does is takes away all the living cells within this organ. It strips it of all the living cells, so it's just an empty, hollow vessel. Just the pure remaining structure is there. And then, as a result, and then after we've done that, after we've stripped it of all the cells, we reseed it. We put in healthy living cells, or progenitor cells, and we'll get into what that means in a little bit. And we put that into this empty scaffold, this empty vessel. Then they start repopulating this organ, and then you have a healthy organ at the end. So before we move on, does anyone have any questions? And I'll stop for maybe like I'll stop for maybe like 10 seconds and see if anyone has any questions. It should be like a fairly simple kind of process. Awesome. Okay, yeah. All right, I'm glad to see everyone is catching on and everything. That is good. Then we will continue with the presentation. So yeah, so that's the fundamental way that that's the basic workflow for how organ engineering works. Let's go a little bit more in depth. You can think of this really as removing and replacing weeds. What I mean by that is, for those of you that garden, or for those of you that own farms, what happens when you have weeds in the farm, right? These are, you know, you can think of them as bad plants, much like cancer or something. We take out the weeds, we take them out of the soil, and we replant healthy plants, and we replant good plants that can grow in its place. So we take out the weeds and we plant something like barley that can grow. That's very, very similar to the process here. We're taking out cells, in this analogy, the cells are weeds. We're taking out bad cells and we're putting in good cells to go and repopulate the organ. So I know the obvious question in everyone's mind is, well, how are you decellularizing? How are you taking away, sorry about that, how are we decellularizing these organs? How are we taking away cells? How does that even work? So there are two main ways we do this. There are physical forces and enzymatic forces. So the physical forces is exposing these organs to high temperature, high stress, high forces, high pressures, and those create environments where normal cells can't thrive and where normal cells start to die out. But the main way, and the most important way that scientists do this, is that they immerse and perfuse. What that means is that they put this organ, so they completely sink it in some kind of fluid, and they make sure that the fluid is able to penetrate this organ and penetrate all the different kinds of cells, both in the internal structures and external structures, with a certain kind of enzymatic solution that lyses all the cells. And we've covered the lysing process before when we were going through viruses, that, you know, it's the whole pop mechanism, that viruses invade your cells and they keep invading until they pop. Likewise, they're just these kind of enzymatic solutions melt away all these cells that are within your organ, so that you have just the hollow vessel left. And I shouldn't say hollow vessel. The internal structures are still there. You still have all the flaps, the ventricles, and the heart, for example, but there's no cells populating it. So you can think of it as just a bare scaffold. And these are just close-up pictures. So in our case with Mia, we would be taking one of her kidneys, and you can survive with one kidney. We can take one of her kidneys, we can decellularize it, so just this scaffold thing, and we can repopulate it with new cells. And when I say scaffold, and when I say scaffold, I want us to conceptually think of it in terms of like a house. So obviously this house I've put in front of you is a very ugly looking bad house. I literally searched up ugly bad houses on Google to go and get this picture. So if we ever have a housing renovation project, what do we do? If we have a bad house that has bad structures, that, you know, the windows and everything is broken, what is the first thing we do? We tear it down to its very scaffold. The scaffold is like the underlying foundation of the house. So we don't, like, you know, completely take away the house. We strip away the walls, we strip away the windows, and we put it down to its bare bones, the bare scaffolds. And that's very similar to the process, almost exactly similar to the process of decellularizing organs. We're taking away all the defunct structures, all the broken structures that aren't working, all the cells that are dying. In the kidney example, all the cells that have cysts on them and that don't work anymore, that don't function anymore, and we're taking them all away. So the basic structure still remains. So we still have the underlying tissues within the kidney, all the underlying complex structures. There's just no cells populating it. And it's a weird concept, which is why I just keep bringing it up so many times, that this kind of structure here is similar to this kind of scaffold structure here. So it's a very kind of interesting kind of process. And for those of you wondering, I think I'm getting some private messages in the chat. Yes, this is actually how the scaffolds look. So I didn't edit anything through image editing or anything. In real life, this is exactly how they would look. If I took your heart and if I stripped it of all its cells, it would look exactly like this. Because what gives it its red pigmentation is the cells within your tissue, is the blood that flows through it. So when I kill all the cells and I strip all the cells away, this is exactly what your organs look like. It's a very cool process. So just so I can ensure that everyone absolutely understands this process, how does decellularization go and fit into this overall picture? So we're going to take this is a cadaveric organ. A cadaver is like a dead body that someone donates to science. So if you're ever a medical school student, you operate on cadavers as practice. So they were real life people who, when they died, instead of choosing to be buried, they donated their body to science. So medical school students can study their kidney, can learn from it, and can be better surgeons if they practice on living patients like Mia. So in this case, you know, we're taking an organ that is either not working or is a cadaver, so it doesn't really belong to anyone living anymore, and we strip it of all the cells. So we can think of this defunct organ, we can even think of this as Mia's kidney. You can think of it as this bad house, right? As this broken house with bad structures. When we decellularize it, when you strip it of all the cells within the organ, then you can think of it as the scaffold, as when we tear down a house. And then when we repopulate it with proper living cells, you can think of it as when we rebuild the house and we put in nice granite countertops, when we put in nice walls, nice bathrooms, nice windows and ceilings and everything. That's this whole process that we go from something that's bad, something that doesn't work, the bare bones foundational structure, and then we repopulate it so it actually works into a nice looking house, nice functional organ. Can STEM be applied? Can STEM cells be applied here? I'm so glad you asked that. I'm so glad you asked it, Dwena. That's actually exactly what I was going to get into. That you might be wondering, how are we repopulating these cells? How are we repopulating this heart or this kidney? We're actually using stem cells, and that's something that I want to recap from last time. That essentially what we're doing is, last time we studied iPSCs, induced pluripotent stem cells. We're taking cells that have yet to differentiate. We're taking progenitor cells, early versions of blood cells, early versions of kidney cells, and we're putting them into scaffold. So in a way, it's like you're developing a new baby all over again, right? When a mother develops a baby in her womb, it's like all these early structures of early blood cells and early kidney cells start developing in the child. And as the pregnancy carries on, these cells keep on differentiating and developing actual structure. Then you go from something that's like like no kidneys at all, no heart at all, to like an actual functioning heart within the child. It's a miraculous, beautiful, beautiful process. And that's what we're taking advantage of here, that we're using human stem cells. And in many cases of organ therapy, this is a very new frontier of science. Like, this isn't fully developed yet. So it's estimated in the next 10 years, we'll have more developed, more advanced mechanisms of organ therapy. But in this, if any of you can do research into this, you'll see that it's very beneficial that if we took Mia's stem cells, or if we took Mia's, saliva cells or hair cells or whatever, made it into an iPSC, made it into an induced pluripotent stem cell, and then we're able to repopulate, an organ like her kidney, then that would be amazing. Because then she doesn't have to run the risk of rejecting an organ. She doesn't have to run the risk of, oh, is the blood type going to match? Is the protein serum marker going to match? Is my body going to reject a new organ? And that's a very, very big problem. And she doesn't have to go through that, because if you're using her own cells, then you completely bypass that process. And her body knows, it's the body cells. So it's a very, very amazing process. So does anyone have any questions on that? Okay, good. And this is just another picture I wanted to show you all to kind of walk you through the process of how I would do this with a heart. So again, it's the exact same process I walked everyone through. It's just a different organ, just to give you a better appreciation for how we can, engineer organs. So let's say we had a heart that had a defect in it. For some reason, the heart wasn't working. It's a myocardial infarction or some kind of chronic heart disease. So what we would do is that we would hook the heart up to a machine. We would essentially pump in decellularizing detergents. And what these would do, exact same thing. They would strip the heart of the cell, and they would only leave the scaffold. They would only leave the structure. So again, all the aorta, all these structures of the tissue and the valves, that's all there. It's just that there are no cells populating it. So as a result, we would then put in progenitor precursor blood cells. So these are like stem cells before they fully differentiated into a blood cell. So I think of progenitor cells as, if you were to give an analogy for humans, if you had like when you have a fetus, right? When the mother's pregnant with a fetus, it's not fully human yet, but it's also like nothing, right? It's kind of like a middle ground state. It's almost a human. It's developing to be human. That's exactly what precursor cells are like. If you want to think of it in terms of adulthood, teenagers, they're in this middle ground between childhood and adulthood. That's exactly what these progenitor cells are doing. They're not fully blood cells yet, but they're not as undifferentiated as stem cells. They're the precursor, something that gives rise to blood cells, something that gives rise to heart tissue. So we can inject that within the heart. We can inject that within the ventricles, inject that within the blood vessels surrounding the heart, and then they repopulate the heart. And all you have to do is give it an electrical shock, and the heart starts beating because all the structures are back there. And some of you may know this, so this is more of a comment for advanced division students, but what makes the heart beat and regulates the AV node, the atrioventricular node, and the sinoventricular node is the Purkinje fibers. So they're located around here. So again, these are electrically stimulated, and they are, again, they are something that the stem cells can give rise to. So it's a really amazing, really fantastic process. So, and these are real life pictures of what this would look like for the heart. So this is a diagram, but this is a real life picture of everything. But this is what the heart looks like when it's decellularized. So after I've stripped the heart of everything, the structure kind of collapses inwards because it doesn't have any cells to support it. And then when we repopulate it, so we sink it in a solution that has all these stem cells, that has all these growth medium, and then the cells kind of grow on top of this and grow within the heart. And that's how we have our end result of an actual human organ. So I'm going to stop for 10 seconds if anyone has any questions on this. So again, I'll pause for 10 seconds. Please type any question in the chat. I want to make sure I'm not skipping over anyone. Awesome. I'm glad everyone is enjoying this and getting it. Why is the aortic valve shut? Well, Jamina, that's a really good question. So the aortic valve is shut because we don't want the let me see if I can get a better picture. One second. The aortic valve is shut because we don't want the let me think of a way to. So if you know the way that the blood flows in the heart, when it comes in through the vena cava here, and then it goes through the aortic valve, through the aorta, and goes to the rest of the body. That's the circuit the blood goes through to deliver blood to the rest of the body. But here, we've isolated the heart from the human. So I should clarify, this entire process, obviously, the heart is outside of the human body. So we don't need the blood to be delivered to other organs, because if we don't shut the aortic valve, all the fluid we're going to put in the heart is just going to leak out and just go out. So we might not decellularize all aspects of the heart. So we're shutting, the aortic valve shuts here, so the blood can actually absorb all the detergents that we're putting through it, and it stays within the heart to be able to decellularize everything. If you were a beginner division, you didn't understand what I said, don't worry about it. It's more intricate mechanics of the heart, and we actually have a lesson within a week or two on mending broken hearts, on how we do cardiac engineering and mechanical valve engineering. It's super exciting. I personally, I really enjoy it. So I think it'll be exciting for a lot of you as well. But Adwina, that was a really good question, and I hope I answered it. So to kind of recap, the aortic valve here shuts because we want all the solution that we're putting in the heart to decellularize it to stay in the heart. If this didn't shut, all the solution would go outwards and just leak out of the heart, right? It's like if I was trying to inflate a balloon, but I popped a hole in the balloon, right? Then the balloon's never going to inflate, right? But yeah, but that was a really good question. And if anyone has any other questions, again, definitely type it into the chat, and I'll get to it when I see it. And yeah, and that's kind of the story of how we can develop new kidneys. And again, like I mentioned, these are developing frontier areas of science that scientists don't have 100% perfect kidneys through this process yet. So for all of you, the challenge for all of you that are college students and high school, secondary school students that are going to be the scientists of tomorrow, these are the kinds of technologies and the kinds of bioengineering modalities that are going to be hot and big when you are then scientists in 10, 20 years, because these are really going to be perfected by science in the next 10 years. So right now, there's a host of problems, and we'll review this in the advanced division with the vasculature within these organs, with making sure that they are biocompatible when we put it back into the individual, back into the patient. And these are issues that scientists are trying to address in the status quo. But it's a really interesting area of science. And if any of you are interested in this kind of research, please reach out to professors, search up, kidney engineering. If you're interested in, let's say, the liver for some reason, or the pancreas for some reason, because maybe you have a history of pancreatic disorder or pancreatic cancer in your family, you can search up, pancreatic engineering research professors, and you'll see a host of results from multiple good universities. And you can approach professors, you can email them and say, hey, I was really interested with how this process works. Can you tell me more about it? Can you give me papers? Can you help me do more research? And that's how we link you guys up. I'll be getting into that process a little later as well. And next, I want to cover bioprinting. So before I even cover bioprinting, I just want to make a quick observational note that over here we have the big field of organ engineering. Bioprinting is a subset of organ engineering. Right? Bioprinting is a way that we can make scaffolds for organ engineering. And we'll get into that process literally in the next slide. But just remember that we have the overarching field of organ engineering. Under that, we have bioprinting, and under that, we have BioInk, which we'll get into. You can think of this as we have a continent. Within continents, we have countries. Within countries, we have states. Likewise, we have organ engineering. Within this, we have bioprinting. Within that, we have BioInk. So I just want to make sure that that is conceptually clear for everybody. So we're going to start off with another case study here. And this time it's going to be a case study of Jonathan. So Jonathan was a lifetime musician. Jonathan was amazing at the saxophone from the moment he was a little kid. He grew up with not well-off family, but he always got through with his saxophone. He'd always play through the hard times. He would play at his school, and then he started being a very high-class musician. So he lived a full life. He toured the world, played saxophone, and played at multiple recitals, went all over the world. And then one day he decided to settle down and start a family. So then Jonathan, right now he's a respected member of his community. He's a devoted father and grandfather of 2 beautiful children. And he teaches saxophone, and he teaches saxophone and how to play musical instruments to lots of kids locally in his community. But one day, Jonathan had a very peculiar experience. Just from getting to his bedroom to his bathroom, he was out of breath. He couldn't breathe. His chest was hurting. His heart was beating like he just ran a marathon, but he just walked 10 steps. He couldn't catch his breath. He was getting dizzy. He was feeling numbness in his left hand. And then he realized he had to go to the hospital. So when he went to the hospital, the doctor informed Jonathan that he was in need of a new heart, and his heart had a chronic disorder, and it would stop working for him soon. So in the meantime, they could give him blood thinners, they could give him medication. That was a bandage on a broken bone, right? That wasn't a permanent fix for him. What Jonathan needed was a brand new heart. But here comes the issue. For Jonathan, getting a brand new heart isn't very easy, because Jonathan now crossed the threshold of 65 years old. He's a grandfather after all. And here's an issue with organ donations. If you're above a certain threshold, if you're above 65 years old, then that puts you on a lower priority list for getting a new organ. Because from the organ donation transplants mentality, someone that's younger might be able to live more life with the heart. So it's unfortunate. And it's even more unfortunate that as every single day progresses, Jonathan's condition might get worse and worse. He might be susceptible to a heart attack one day, not even know it. And this means that we have to ask ourselves, what kind of quality of life is this? He's reliant on the hospital almost every day, almost every week. He's reliant on his medication, on his healthcare providers, constantly getting his blood pressure checked, constantly getting ultrasounds and CTs of what his heart looks like, what kind of defects are there, and that takes away from his quality of life. If Jonathan only has a year or 2 more to live, then Jonathan doesn't want to spend it in a hospital. He wants to spend it with his grandkids. He wants to spend it playing saxophone. He wants to spend it working with the beautiful kids that he got the opportunity to work with. And that's the unfortunate reality of hundreds of thousands, if not millions of people around the world. Heart disease is the #1 killer of people in the United States, even beyond cancer. And that's something that's very, very glaring and very, very overlooked for patients like Jonathan. So the question we need to ask ourselves is, well, what can we do for patients like Jonathan? And one thing that we can do is obviously organ engineering, and we can populate scaffolds. We can decellularize organs, and we can populate those scaffolds with stem cells. But what's going to happen when we take out Jonathan's heart? Right? Jonathan's not going to survive without a heart unless we put him on some kind of ventilator support or some very intensive surgical support. And that's going to be a challenge in and of itself. And the entire process of organ engineering right now is incredibly expensive. And for those of you that are interested in this space, that's also a place that requires innovation. That even if organ engineering is invented, ask yourself, what can you do to make it cheaper? That could be an invention, that could be a life-saving solution in and of itself. But for patients like Jonathan, we can avail something called bioprinting. So bioprinting is very, very similar to organ engineering, but it occupies the space and occupies a process in organ engineering of developing the scaffold. So bioprinting, whereas organ engineering that we just covered takes an actual organ, right? An actual kidney, like we actually took Mia's kidney in that other case. In this case, we actually took out a living heart and we decellularized it. In organ engineering, what we're doing is we're creating our own scaffold. So we don't need to use another organ. With bioprinting, we're creating our own scaffold and printing our own scaffold. So this process is laid out for you here. But before you even look or read into it, listen to what I have to say conceptually. So if I want to print a 2-dimensional picture, let's walk through the steps of what I need to do in 2 dimensions before we go into 3 dimensions. So let's say, I'm a student, I'm an MD candidate at Brown, right? I'm a student at Brown. So when I go off to college, I want a picture of my mother when I go off to college, right? I don't want to forget her. I want to see a picture of her every day when I wake up. How do I go about doing that? So the first thing I need to do is I need to take a picture of my mother on my phone, right? I need to have a digital copy of what my mother looks like in 2 dimensions, and the X and Y dimension. So then I have a digital copy of my mother's face. Then what do? I print out that image. I go and I link my phone to my printer, and then I click print on my phone. And then what the printer does is that it translates every single pixel and the position and color of every single pixel of the picture of my mother's face, and then prints it out in 2 dimensions on the printer. So when the printer is printing my mother's face, it knows that on this coordinate, on this specific coordinate, print red for her red dress. On this specific coordinate, print black for her black hair. And then that's where it goes back and forth, is that it translates the pixels that I have on my picture to a 2-dimensional physical image. And that's a physical image I have right in front of me that I can take with me to college. So that might seem tangential. It might seem like I'm trying to bore you with a life story of mine, but I promise I'm not. The reason I give you this story is that it's very, very important conceptually to understand how 2-dimensional printing works, to understand how 3-dimensional printing works. So the process of 3-dimensional printing is incredibly similar, except now we introduce a z-axis. So what do I mean? So let's look at all the correlates as I walk you through the steps. So let's say I want to 3D print a scaffold for the heart. How do I go about doing this? So the first thing I need to do, right, in my mother's example, if I wanted to have a picture of my mother, the first thing I need to do is take a picture of my mother. I need to translate what physically exists of my mother's face and translate it into 2-dimensional plane of pixels on my phone. So likewise, for the heart, if I want to recreate the heart, when I take a picture of it, I need to create a 3D scan of it. So again, it's basically like a picture, but instead of 2 dimensions, it also has a 3rd dimension, which shows me the depth of how deep is the heart, what kind of intricate structures of ventricles are there, how wide do certain parts need to be. So I have a 3-dimensional scan of the heart, and then I have that in my computer. So a CAD model is like a computer animated design. It's a computer model of what a heart would look like digitally. So then I have that digital copy. So again, in my mother's example, what did I do? I printed that picture of my mother's face. So likewise, what we're doing is that we're connecting a 3-dimensional CAD model, a 3-dimensional model of the heart, to a 3D printer. And again, a 3D printer, it sounds complex. A lot of people talk about it. It's genuinely just a normal printer, but if the way a normal printer goes is this way, right? 2 dimensions. So it prints, it prints my mother's face or whatever. But in 3 dimensions, it prints upwards. So it has 2 dimensions, and it also extrudes into the z-axis. So that's all it is. It just prints in 3 dimensions. So I have my heart scaffold that's printed through a 3D printer, and as a result, at the end, I have a 3D bioprinted tissue product. So that's the fundamentals of how bioprinting works. And I'm going to go into each of these individually. And there are definitely multiple questions that have to be answered along the way. How are we scanning the heart? How are we getting this into a 3-dimensional scan on our computer? How is the computer communicating this to a printer? How is a printer able to print this, right? What is the ink that the computer uses, that the 3D printer uses? And that ink is known as BioInk. And that's something we'll get into in like 4 or 5 slides. So I'm just introducing the idea here that the way that my printer needs an ink refill, right? I have colored ink, I have black and white ink to print, a picture of my mother. Here, I have BioInk to print the scaffold of the heart. And then I have a working 3-dimensional 3D model, 3D tissue model of the heart. So again, let's answer all these questions sequentially in order. So firstly, let's look at the first thing we need to talk about: scanning. So this is a kind of this is for creating a model of the ear, but I just wanted to include this because it's more traditionally in line with what we think about scanning. There are very highly advanced cameras, and they kind of shine this light, and they kind of go around you, they go around the person, and they create a 3-dimensional model, and they take pictures from every single angle, and they kind of overlay to see, can we create a model? What this looks like for the heart is that we actually use CT scans, computerized tomography, and I'm just going to give everyone like a quick 90-second background on how this works. What CT scans, computerized tomography scans, do is that they're basically layered X-ray scans. And the best way I can kind of explain this is that they take a bunch of segments. So they take a bunch of pictures of segments of the heart, and they combine them together. So I am reminded of stop motion pictures a lot. That if you take a picture of me doing this, of me doing this, of me doing this, of me doing this, this and this, and you put all those pictures back together, and it looks like a video where I'm waving my hand. That's how cartoons were made in the old days and everything. So that's something I want to drive home to. That obviously, you know, this is obviously a stop motion picture. These yarn products didn't start dancing or jump roping on their own. Someone took a picture of these yarn products in every single position, and when you put them together, it looks like it's a video of them like jump roping and them playing together. So you know, it's this combined process that looks super cool and super exciting. So that's exactly what we do when we're trying to do it with the heart. That we take a bunch of layered pictures of the heart, right? A bunch of individual layered pictures of the heart. And then we combine it together, combine these 2D images together. We're able to create a 3D model of what the heart actually looks like, and see and visualize a 3D model of all the intricate structures. Where are the ventricles? Where are each nodes? Where are all the septum? Or septa, where's the aorta and all the other blood vessels that lead into the heart? We can create a really advanced model just like that. So it's a very, very interesting process. So now let's recap and backtrack. So we had a patient's heart, we've scanned it, we've done a CT scan. Now we have a solid 3-dimensional model of what our heart looks like. So now we have it virtually. So then the next question is, how do we go from the virtual to the physical? So again, the communication process was the second question, right? How do we go from a digital image to something that's physical? Well, the answer to that is the exact same way that our printers work at home. So by the exact way that our printers, when it translates a digital signal of my mother's face on the phone and matches each pixel to an actual physical color it's printing on a piece of paper, that's exactly what happens when we're printing the scaffold. That except now it happens in 3 dimensions. So the computer basically takes this digital visualization of the heart and then is able to print a scaffold according to where each 3D pixel is. So again, remember that this heart model is in 3 dimensions. So it tells them the XYZ coordinate of where every piece of the heart is. So when the computer is copying it, when the printer, is copying it, it knows exactly where to lay down each piece, where to lay down each cell. And that's how we get scaffolds. So then the next question I asked you all is, well, what is it printing with? What is this material? Well, that is BioInks. So the way my printer prints with printer inks, this is BioInks, because everything sciencey has the word bio in front of it apparently. And this process and this and these BioInks are essentially what allows us to print this scaffold and print what this preliminary version of the heart looks like. So in Jonathan's example, we're literally printing like a new heart for him from scratch. And that's an amazing, amazing process. So I'm sure a lot of you are wondering, well, physically, what is BioInk? Well, it's a kind of hydrogel material. So think of it as a kind of intermediate between liquid and solid. And toothpaste is a good example of this, right? I'm assuming that everyone here brushes their teeth. So you're aware of how toothpaste is both has liquid and solid products at the same time. That's different from the material used here. But conceptually, I want you to think of it like that. It's kind of intermediate between fluid and solid. And that gives us a benefit of it's malleable as a fluid, but it can also hold its state like a solid. Another good example is it's like clay or Play-Doh, where I can mold it to be in whatever shape that I want it to be. And the actual contents of this fluid are live cells and biopolymer gel. So these live cells can help to create places for the actual these cells create places for the actual stem cells when I repopulate the scaffold to thrive in. And the biopolymer gel is essentially made of materials like collagen, gelatin, silk. So these are the framework. These are the scaffolds that help to hold these cells in place. And very often, these biopolymer gels are made so they can dissolve away if I put it in liquid or if I put it in the human body. And some of you, and before I get into that process, I just kind of wanted to show everyone this progression from scanning like an organ like the ear, making a 3D printed scaffold, and then repopulating it. So remember, what I'm bioprinting here is the scaffold. So I'm printing the scaffold. I'm not printing the finished product. So I just want everyone to be aware of that. And for the record, this actually happened in real life. Scientists at Cornell University, it's an Ivy League university, essentially did this exact process where they scanned someone's ear. And this is an actual picture from when they were doing that. So they were actually scanning this man's ear to recreate. And then they were able to recreate the ear, repopulate it, and they made a human ear. So this is already happening in the world right now. So I'm not talking about science fiction. I'm not talking about theoretical. This is actually happening. And that's amazing, right? Like that we're making organs of humans. Like that's insane, right? And to go a little bit, to delve a little bit deeper into the process of how the scaffold works, I'm going to walk everyone through it right now. This diagram might look complicated, so just bear with me. I promise it's very simple if you just listen to what I have to say. So essentially, what we're bioprinting here is a scaffold. So this scaffold is this 3D printed filament network. So what we're doing after we create this filament network is we're encapsulating it in a liquid network of living cells. So the image I want to show everyone is this one, that once we have the scaffold, we're dipping it in this entire liquid solution of live cells, live stem cells, and growth medium. And that's exactly what we're doing here. So we're taking the scaffold like here, and we're encapsulating it in this actual solution of live cells, live stem cells that then populate the network. And then what happens is that we can dissolve away the network. So based on what kind of scaffold you use, what kind of BioInk you use, if you just pour water on it, things like gelatin or the silk basis will dissolve away. And this is very similar to I think drug pills. When we take medicine, when we ingest medicine, similar conceptually to when we ingest medicine. So let me kind of clarify what I mean by that. So when any of us take medicine, right? When we have, let's say we're taking medicine for digestion issues, or we're taking medicine to, I don't know, have healthier heart. What we're actually needing is the actual drugs within the medicine, right? The actual capsule doesn't help us at all. The capsule is just a way for us to deliver the drugs to our stomach. The capsule is biodegradable. So if you ever take these medicine capsules like probiotics or something and hold them underwater, the capsule starts dissolving. And that's exactly what happens when I swallow this capsule. As it goes through my saliva, as it goes through my esophagus, the capsule dissolves away. And when it goes to my stomach, all that's left after the capsule dissolves away is the actual drugs, the actual medicine that I need to have for digestion or heart issues or whatever. So that's very similar to what we're doing here. That we're using this scaffold design to show us a structure of where should each cell go? Where should the intricate structures in our cell go? And then when we put it inside of the body, inside high temperatures, inside liquid, inside the blood, then it's biocompatible such that it dissolves away, it's biodegradable. It degrades away within the body with blood or with water or whatever, and all that's left is this population of cells in the exact order that you want it to be. And I have this model of the house here to give you this analogy. And as you all know, I love analogies, and this one I particularly am fascinated with real estate. So just bear with me here. If I want to create a house with 2 stories, and I want a kitchen on the top story, and I want to place my chair right here, how on earth would I do that if I didn't have this scaffold? So again, to understand the importance of something, we need to understand what would happen if it didn't exist. So imagine I was building a house and I wanted to put a kitchen on the 2nd story. Well, how would that kitchen exist on the 2nd story? If I threw my kitchen chair up in the air, what would happen? Obviously, it would fall down because of gravity. So the exact same thing here. If I'm building a heart, and I know that I want, this top ventricle to be over here, and I want this top ventricle to be over here. I want this aorta to be over here. I want this kind of, left ventricle to be over here, or this atria to be over here. How is that going to happen when gravity exists, right? If I was just to put these cells in a medium plate, they would just grow in 2 dimensions and start growing up, and I wouldn't get a heart structure. I'd get something that isn't a heart. That's something that collapses. So that's why we put in the network temporarily to help the cells grow around, and then we can remove that. Then we can remove and dissolve away that network the same way we dissolve away the capsules on medicine. Another way to think about this, another analogy that I just thought of, is when you're growing certain kinds of plants. Certain kinds of plants, they can't grow without support. So you have to put in like a big stick or a big kind of straight edge or like a ruler or something. And the plant, like vines or something, would grow around that straight edge, and they would grow upwards with the support of the straight edge. And then you can take away the ruler, you can take away the stick or whatever, and then the plants can grow as normal. So you have that straight edge there, so they have a fundamental structure to grow around, something to give them the shape they need, and then you can take that away. That's, again, the exact same thing here. We're putting in this initial scaffold, so the cells have something to grow around. The cells know, well, over here at the top is where I can be ventricles. Over here at the bottom, where I can be, atria. Over here at the bottom is where I can be ventricles. Over here at the top is where I can be the aorta or whatever. And then the cells assume that position and assume and differentiate into the proper structures based on the position. And that's actually a very amazing thing. And so amazing that I think it warrants me saying it one more time. That based on where the physical position is, these stem cells are so intelligent that they know what to properly differentiate into. So if they're at the bottom, they know to differentiate into a ventricle versus at the top into an atria. So our cells are ridiculously intelligent, and it happens every time, you know, a new child is born. That when based on the position of where the child is in the chest, where the stem cells occupy, they know what parts of the heart to differentiate into, or whether to differentiate into the heart over here or the kidney down here. It's a really fantastic process. And I just kind of had this slide here to illustrate to you that, yeah, once you put in stem cells, right, it's like a cell-less scaffold. They start developing these intricate structures of blood vessels and of tissue networks and everything the second we repopulate them. So over here, we're using mesenchymal stem cells. It's a big word, but it basically just means stem cells dissolved from the bone marrow, derived from the bone marrow. So M for marrow, M for mesenchymal. That's a nice way to remember it. But we're using stem cells from the bone marrow from a child or for the human. We're repopulating an organ with it, and you have all these wonderful, fascinating, complex networks that can grow. And that's really an amazing process. So that's essentially most of what I have for you all today. I think I have another case study that I can cover with the advanced division, unless everyone wants to have it. Wants to hear it. It's more stuff about essentially lungs. The next case we're going to cover is of Timothy. So Timothy is a hardworking father. He's lived a long life. When he was a kid, you know, Timothy was a party type. He would do lots of drugs. He would smoke and everything. Ever since then, he's been weaning off his drugs, but he has a bit of a smoking history. And he recently found out that he was diagnosed with terminal lung cancer. So terminal lung cancer is extremely serious. It affects hundreds of thousands of people around the U.S. every single year. It's an extremely devastating disorder. So what happens in terminal lung cancer is that your lung tissue, over here you can see scans, you have tumors growing in your lung tissue that make oxygen exchange extremely difficult. So they basically rupture your lungs, they take up space in your lungs, they make the blood oxygen exchange process difficult because they destroy a lot of your alveolas and all of that. So it's a very painful process. So the status quo, getting lungs is hard because I can donate a kidney, sure, like to someone like Mia, like our first case scenario. I can donate my kidney and I could be fine with one kidney, but I can't donate a lung. I will not survive with only one lung. So that's a very difficult place to be in. But here's an issue with bioprinting lungs. So because of the nature of lungs, they have three properties. So they have wide surface areas. If you kind of splayed out the surface area, I think they would take up like, I don't want to misquote, it's like 30 meters of square feet or like 3 miles or something. It's an incredibly large amount of space that your lungs take up. So because of that, they have very thin membranes and they're very intricate structures. They're very intricate structures. And because of that, they're incredibly difficult to virtualize and actually print out through a 3D bioprinter. They require extreme precision that modern science doesn't have. And also, they have viscoelastic behavior. So your lungs, I go, right? When I'm breathing in and breathing out, my lungs are expanding and they're contracting. They're expanding and they're contracting. So the scaffolds we build through bioprinters aren't that expandable. They're not that elastic that can allow us to breathe as much as we want to. So these are very kinds of extensive processes, very kinds of intricate processes. And when we try to make these with synthetic scaffolds, we don't have enough control over these micro-vasculatures, these thin, small, intricate processes. An equivalent of this would be if I took a picture on my phone of the starry night sky and expected to see the star, like expected to see every single crater in the star, every single crater in the moon, or every single aspect of the planet or something in perfect HD 3 dimensions. That's just never going to happen. My cell phone is never going to be that powerful to recreate a good image of something so far away. The best I get is some kind of blurry dot. So this exact same thing here. If I want to recapitulate, basically recreate these kinds of intricate hierarchical structures of these alveoli, which basically conduct oxygen exchange with the blood, I'm just not going to be able to do it because they're so tiny, so difficult to do. And also and that gas exchange process gets completely screwed up. So that's where we have the initial process of not 3D organ printing, of bioengineering, of engineering organs. So this is exactly very similar to what I discussed with the Mia example initially. That with Mia, she had a defunct kidney, and we were decellularizing the kidney, and then we were repopulating it with stem cells that were able to regrow and make it a proper kidney. So that's exactly what happens here, where this was a trial that was done in rats and then was done with an actual human lung. So I'm going to walk everyone through the process of what happens in rats. And again, it's all going to be stuff you know. I'm just applying it to a different organ. So I hope everyone can kind of have an appreciation for how you already understand this process. So we're going to take Timothy's lungs, right? Timothy's our patient who has lung cancer. We're going to take his lungs and soak it in a solution that decellularizes it. So we're going to soak it in an enzymatic solution with high pressure, high temperature. That's going to kill all the cells. But the intricate structures are still going to remain. So these structures that were originally in Timothy's lungs, all these alveoli, all these kinds of branches and everything, these exist. These will continue to exist. The only reason they function in the body is because we have cells that populate the vasculature and that help conduct oxygen exchange. We have blood that goes through them that helps deliver CO2 to the atmosphere, and we have molecules like hemoglobin that capture oxygen, put them back into our system. So that's why this structure works. But without these cells, these are useless. But they still have the intricate kinds of processes, intricate kinds of capillaries and membranes that we want. So now that we have an empty, hollow organ, we can do two main things. So we can either, and they're just different orders of doing the same process. So either we can cut open sections of the lungs. We can cut open sections of the lungs and seed cells. So this is an easier process to seed cells if we're not confident that they can, the stem cells can occupy the hierarchical nature of the organ. So we can cut open like a two-dimensional slice and we can put in new stem cells. Or another thing we can do is we can take the entirety of the lung. We can put in these progenitor cells. We can put in these stem cells. We can take sections of that. So we can take sections of the lungs that have already had stem cells deposited all over them. And then we can culture them in some kind of media. So again, these are two different ways of doing the same thing. So scientists are right now conducting tests to see which one has higher efficacy rates. Is it better if I take just sections of the scaffold and then populate it with cells? Or is it better if I populate the scaffold and then take sections of the populated scaffold and then culture it or something? So it's like, again, two ways to do the same thing. And scientists are trying to figure out which one is more effective. I think there's some evidence suggesting that this way, seeding the cells and sectioning it is more effective, but that's still like developing in an area of science. And again, this is functioning like if I'm making my sandwich and it's functioning like if I'm having breakfast and I put cereal first then milk, or milk first then cereal. That's the difference. In the end, you're getting a similar product, but maybe in one way it's more effective than the other. So that's that. So then once I'm able to put together the lung and repopulate it with seeded cells, what I'm doing is outside. So ex vivo means outside of the body. And these are two important terms that we should know. In vivo and ex vivo. Ex vivo just means outside of the body. In vivo means inside the body. Vivo, I think, has some Latin roots. I think it means like body, person, so something like that. I don't want to misremember it or something. But ex vivo, ex means outside, in obviously means inside. So when something happens ex vivo, that means that I'm doing it in a lab setting. So this, that means if something happens ex vivo, that means that I have, like that earlier image, let me see if I can find it. That earlier image of, right here, yeah. So this would be ex vivo, that I've taken the heart outside of the body and I'm soaking it with culture, trying to repopulate it. So it would be exactly this image, but instead of a heart, it would be like a lung cell. So that's all ex vivo means. So what happens is that I test it outside of the body to see, does this work for gas exchange? So this is important because I don't know if this lung works yet. So if I put it into a human being, or in this case, a rat, and don't know how it works, the lung might not expand properly. The lungs might have issues with the cellular composition, and then the rat's just going to die. So my patient is going to die. If I put this into Timothy without testing it, obviously there could be disastrous consequences. So the way that scientists get around this is that they test this outside of the body. So they actually hook up the lungs to an oxygen platform. They put oxygen into the lungs, and they see, is the lungs expanding? Can we see oxygenated blood flowing through the lungs? Do we see proper circulation? And then we put the lungs back into the rat or back into the human, and then we see how is gas exchange happening. We close control on what is the blood pressure, what is the O2 saturation, what is the percentage of red blood cells that has oxygen within the blood, and then we see, are there any possible side effects? So edemas are kinds of we don't need to get into that. That's more medically complicated. But these are what's important to know is that these are just medical complications that can arise as a result of organ transplants. These processes aren't perfect. So one thing I did want to direct everyone's attention to is that these are, like I keep saying, novel areas of science. So right now, scientists are looking into grow your own organs with pigs. That scientists have engineered pigs that have very similar organs to humans, and we're testing it on pigs with pig hearts, with pig livers, with pig lungs, and seeing if it works. So this was, I believe, a year or two ago that this came out. So expect more news reports on this happening in the status quo within the next, couple years happening. And when you are all like scientists in college or scientists in graduate programs actually working on this stuff, that's when it's going to be like popular. That's when media is going to be covering it. That's when we're going to be seeing, oh, is this possible? Is this feasible? And just the last kind of thing that I have to talk about issues is creating microvasculature. So I didn't want to cover this in the beginner divisions because this can get a bit more complicated. But a big reason why we don't have three-dimensional organs working is because we can't implement proper microvasculature. That maybe I can get all the different ventricles and valves and cells within my heart to work and function properly, but none of that is going to matter if my heart can't get blood. None's going to matter if my lungs can't get blood. Those cells are going to die out. And the issue is similar to like I said with the lungs before, because these vessels, because these networks are so intricately crafted, because they're so tiny, it's so difficult to reproduce and repopulate. And oftentimes when we're decellularizing scaffolds, it's hard to know, well, where are these kinds of blood vessels? Where are these kinds of veins and arteries? And how are we properly repopulating them? Are we properly constructing arteries that work properly for the body? It's a very, very kind of complex process. So we can make scaffolds for this through PLA, polylactic acid. That's a common kind of, that's a common substance that scientists use in this area. And then we can put in nanofiber conduits. So all that is that the nanofiber conduits are like, how can I explain this? They're like miniature, they're porous miniature strings in a way. They're porous miniature bits that go in to aid oxygen and blood exchange. So these basically go in the arteries, go in the help to facilitate the development of arteries and of blood vessels to deliver blood to the heart or deliver blood to the lungs. And the real challenge here is that how are we able to engraft these microvasculatures? Are we able to engraft these arteries and veins, these small tiny vessels that deliver blood to and from organs into the organs? So the main way that we do that, again, is through 3D printing and bioprinting these microvascular filaments. And we bioprint the scaffolds, and then we populate them with stromal cells and endothelial cells and the specific tissue of like heart cells or lung cells. So endothelial cells is like the internal lining of the cell. The internal lining of like a blood vessel or something is the endothelium. And stromal cells are more like connective tissue. So when we combine all of these, so when we put like the internal lining of like a heart blood vessel plus connective tissue for the heart blood vessels to connect to the heart, plus tissue specific cells like cardiac myocytes, cardiac cells to populate these blood vessels, then we are able to get structures that have capillaries, that have blood vessels, that have lumen, that can deliver blood to and from the heart. I believe someone asked a question somewhere. I will check up on that. Oh, never mind. No question. Okay. So that are able to populate the heart. And in real life, when scientists are making these structures, so they're making them in these kind of, it looks like gelatin kind of structure. It's similar to the image I showed you all with the heart dissolved in all the water. Not dissolved, the heart immersed, sunk in all that red liquid of all the stem cells to populate it. Same thing here. So again, we're creating this scaffold for this vasculature. We're creating the scaffolds for this vasculature. Then we're populating it with actual stem cells, and that's what they look like. So this is like a very zoomed in picture, but these are these complex filaments and networks that can deliver blood and oxygen and capillaries capillaries that facilitate gas exchange to organs. So still in the status quo, having a high resolution, a high level of intricacy for these capillaries is a huge problem. So being able to have accurate blood vessels, because they're so tiny, is very difficult for us. But that's something that scientists are trying to work on. So they're having high resolution, more intricate, more advanced solutions by the day, every single day. And to ensure that these kind of connect well with organs, they give VEGF. So VEGF is a kind of angiogenic growth factor. Some of you may have heard of angiogenesis in the context of cancer. When cancer undergoes angiogenesis, it's when a tumor connects with all the blood vessels in the body, and it goes and is able to spread through your blood. So if I have, let's say, lung cancer, if it undergoes angiogenesis, it basically grows blood vessels, takes nutrients away from other cells, and is able to spread through the bloodstream. So that's obviously a terrible thing when it happens with cancer, but it's a great thing when it happens through our blood vessels, because our blood vessels need to obviously take blood from the body, all over the body, give it to the heart. Take the blood from the heart and give it all over the body. So those are the kind of growth factors that we can give to ensure that these work properly. And obviously we circulate media and different kinds of all these growth factors through ex vivo mechanisms to ensure that they grow properly. And again, this exact same process of we have a cellular scaffold, we scaffold of, let's say, like microvasculature, like blood vessels. We populate it with stem cells, and we dissolve the network away when it comes in contact with blood. That's the exact kind of same mechanism we're using for the blood vessels and microvasculatures as well. So I know that was a little quick, but that's basically all I have for you today for the advanced division. So to kind of recap what we learned was we learned about the issues with bioprinting, the limitations of bioprinting. So if you have something like a lung, it's very difficult to bioprint because it's viscoelastic, it stretches out, you need blood exchange, gas exchange that we can't quite replicate through blood vessels. And then we covered, well, how do we replicate this through blood vessels? How can we create blood vessels by, again, decellularizing and creating micro scaffolds for the microvasculature, creating scaffolds for arteries and veins to deliver blood to and from organs. Some limitations of that because of a lack of resolution, and ways to ensure that it happens better with things like VEGF, to ensure that angiogenesis occurs through the blood vessels to ensure that they can properly facilitate blood exchange. So if anyone has any questions, totally feel free to ask that in the chat, but and I'll wait for about 10 seconds for that. Okay, wonderful. I think everyone's getting it. Everyone's hopefully enjoying it and learning stuff from here. So again, thank you just so much for coming and joining us for the lesson and sticking around for the advanced session. Dorina, yeah, repeat the function of the microvasculature and how they engraft organs. Yes, that's a very important question. Yes, I will get to that right now. Let me get to the exact slide that I think will help you. Right here. Yes. So the process of let me look at your question one more time. So get to the process, repeat the function of microvasculature and how they engraft organs. Okay, so there are two questions implicit within your question. Firstly, what is the function of microvasculature? The function of microvasculature is to deliver blood to organs and to take blood away from organs. So for the lungs, for example, we need something to give blood to the lungs, give blood to the lungs such that there's oxygen exchange. So the way oxygen exchange happens in the lungs is that I breathe in air, there's air in my lungs, and that air through these capillaries, through these alveoli, and all of that, the oxygen basically interacts with my blood. So that's what these tiny networks here are for. So the hemoglobin in my blood picks up oxygen and delivers carbon dioxide to my lungs. That's why when I breathe in, you can take a deep breath right now, and all the oxygen in your lungs is going into your blood, and your hemoglobin is collecting that. And it's also releasing CO2 into your lungs. That's why when I hold my breath in my lungs, and I exhale, it's all carbon dioxide, not oxygen, because all the carbon dioxide from my body has been entered into my lungs and then been expelled out. So that's what microvasculature is for. So this is microvasculature. It's for facilitating oxygen exchange. Microvasculature helps give oxygen to the blood and give carbon dioxide outside. And that's what helps us survive and live. So the way we engraft this into organs is that we make scaffolds. So there's two ways to do this. We can, again, decellularize actual existing vasculature structures, or we can create our own. So we can take polylactic acid, PLA, take nanofibers, so porous nanofibers. So they need to be porous so they can absorb blood and also release blood and everything. And they use an electrospinning mechanism to basically ensure that this is a proper scaffold that can be integrated within the organ. So that electrospinning mechanism is what helps us engraft this into the organ structures. And then once we are able to grow and seed it with stem cells, then they become more sustainable as something that can grow within the network of organs. And then we can dissolve the scaffolds away. We can dissolve these kinds of scaffolds away, and all that's left is the actual blood vessels like the arteries and the veins. But I hope that was helpful, and I hope that helped you